It's a pleasure to introduce Pilar Cossio, who is a Max Planck Group Leader associated with the University of Antioch in Colombia and the Max Planck Institute of Biophysics in Germany. She got her PhD in 2011 in computational biophysics at CISA in Trieste, where she worked on protein folding using enhanced sampling techniques and MD simulations. After that, she did her postdoc at the NIH and the Max Planck Institute, developing methods for extracting structural and dynamical information from single molecule force spectroscopy, Cryo-EM, and MD. And since 2016, she leads a Max Planck tandem group associated with the University of Antioch in Colombia. And she is going to be talking today about Bayesian inference on free energy profiles, I believe. And I think if you want to share your screen, Pilar, see if it works. Yeah. Yes. So take it away.
Okay, so thank you very much for the invitation. I'm very happy to be here. I've really enjoyed all of these sessions, and it's been really encouraging to see people from all over the world, experimentalists and theoreticians, working together and looking for new problems and exciting topics. So I really hope this can continue even after the pandemic. So today I'm going to talk to you about Cryo-EM Bayesian inference of free energy profiles. This is a work that I did in my group at the University of Antioch in Colombia and the Max Planck Institute of Biophysics, and in close collaboration with the Flatiron Institute in New York. So I'm going to start with the motivation of our work, why extracting free energy profiles is important, what can we learn from free energies, and how can Cryo-EM help us extract free energies. Then I'm going to present our theory, a path collective variable for Cryo-EM, and our Bayesian inference methodology for extracting free energy profiles. Then I'm going to present some results for synthetic data where we have full control over all the Cryo-EM imaging conditions, and really we want to push the system and push the method and really ask, when does it work? Under what conditions does our method work? And finally, I'm going to present some results with real Cryo-EM data for a calcium activated ion channel. So why is the free energy important? We think that the free energy ultimately will tell us about the biological function of the biomolecule. So not only having a high resolution, most probable conformation will give us information about how the biomolecule works, but also understanding the metastable states of the system, the transition states of the system, and the transition paths. How these states are connected will tell us how the biomolecule works. However, extracting the free energy is very challenging for like traditional structural biology techniques. And the reason is that, for example, X-ray crystallography, NMR, and SACS are ensemble averages. So basically, the observable that you take is an observable over an ensemble averages, where you have, for example, multiple copies of your protein in a crystal, and then you measure an observable. So Cryo-EM brings us a great advantage because really Cryo-EM is a single molecule technique. And the freezing process is done very rapidly. And so basically you can trap the individual conformations of your biomolecule, and each projection image in principle corresponds to a conformation that is distributed according to Boltzmann factor at the temperature just before freezing. So in Cryo-EM, because it's really a single molecule technique, we have much more information than just an average, and we can use this in our advantage, for example, for extracting free energy profiles. So one of the challenges of extracting a free energy of a biomolecule is that the conformational space is extremely large. So if you have a protein with, say, 10,000 atoms, you have 30,000 degrees of freedom. Your conformational space is really, really large. So one of the key questions to have a comprehensive description of the free energy is to reduce the dimensionality. So you're reducing your 30,000 dimensional system into something along a couple of dimensions, but that is human interpretable and that makes sense. So what has done so far in Cryo-EM? So one of the first milestones was this paper by Joachim Frank's group, where they used machine learning and diffusion maps to make a low-dimensional projection of the free energy landscape and understand how the cycle of the ribosome works. So more recently, a couple of works have used principal component analysis, and the 3D maps are reconstructed using, say, traditional cryo-EM reconstruction techniques, but then they perform PCA over these maps, and then they can have a low-dimensional projection, the couple of first principal components, and extract a free energy landscape over using PCA. So something interesting is that there are really just a handful of papers that have used Cryo-EM data to extract free energy. And the main reason, well, we all know it, is that basically Cryo-EM has gone through the resolution revolution, and nowadays it's amazing the atomic details that we can obtain in Cryo-EM maps. However, I just want to say that there is still room for extracting more information, for understanding much more these biological systems using Cryo-EM. And there are several issues that and questions that remain to be answered. So the first one is it possible to recover really the free energy from Cryo-EM data? So instead of having 3D conformations, you have 2D projection images, and you have a very low signal to noise ratio. So under what Cryo-EM conditions can we really recover an underlying free energy profile? The second question that we have is, okay, so now we're extracting free energies. How do we weigh the particles? So we know that in traditional Cryo-EM reconstruction techniques, a huge percentage, a very large percentage of the particles are classified out, and only a small fraction of these are used to reconstruct a high dimensional structure. So in this case, how do we weigh the particles? How do we filter out the particles? What do we do with them? And sometimes PCA and machine learning methods are not always ideal, or are a bit cumbersome to really apply. So I come from the MD community, from the molecular dynamics community, that has developed many methods for extracting free energy surfaces dealing with large conformational variability, where you have thousands of different protein folds, about millions of different conformations. So how do we extract the free energy, which means reduce this large conformational space onto some low dimensions that we can understand? And along these lines, in the MD community, are collective variables. So for a collective variable, the projected free energy profile should be an accurate representation of the multidimensional free energy landscape and also the dynamics of the system. What does this mean? This means that for a collective variable to be good, you should be able to separate the welds, to separate the metastable states, identify these metastable conformations, but also identify where the transition state is, and identify, for example, the activation barrier height. And all of this with respect to the should be an accurate representation of the multidimensional free energy surface. However, one of the limitations of collective variables is that this is not always true. Not all collective variables are good, and it's actually difficult to prove that your collective variable is indeed adequate. Nonetheless, collective variables have proven very useful for understanding diverse phenomena, from like soft matter physics to, for example, nucleation, to protein folding problems. They have been used a lot in a large amount. Okay, so this is basically our motivation. And now I'm going to present to you the theory that we've been developing. So this is a work in collaboration with Alex Barnett, Bob Carpenter, and Eric Thiede from the Flatiron Institute, and developed by two of my master's students. So the first idea that we want to present is to develop a path collective variable. And this is inspired by the methods developed for the MD community, in particular by Michele Parinello's group, where you have your multidimensional free energy surface. So say that this is your large conformational space. And then what you define is you're going to define a path, a 1D representation of your system, but that involves large conformational changes and complex conformational changes that can take you, for example, from one state like an open ion channel, and you go smoothly through the closed state of the closed ion channel. And so basically what this path represents is a continuous and almost semi-continuous changes, conformational changes that go from one relevant state of your system to another relevant state, and that can pass through multiple relevant states of your system. So what you're doing is you're taking your large conformational space and you're really projecting it onto a complex path, but that tells you the relevant changes of your system. So here M is going to define a node along the path, a 3D conformation, one of these 3D conformations along the path. Omega is going to be a Cryo-EM particle image, so a 2D projection. And G is going to be our free energy profile along the path. So say that we have a set of particle images and we have defined our path. What we want to develop is a method that's going to calculate, assign a probability of a free energy profile along this path, given the set of images. And so we're going to propose this Bayesian formalism, the Cryo-BIFE formalism, to assign a probability to a free energy profile given a set of 2D images. So for a single image, we're going to define the probability of that image given a free energy profile G as the sum over all the models in the path. So this is a discrete path, but you can have many models along the path, of the probability of that image being generated by that model times the probability of the model be given the free energy profile. So the first term here, what we're going to use is we're going to use a Bio-EM formalism. This is a method that I developed together with Gerhard Hammer. And what this does is it calculates the likelihood of a single particle image being created, being generated by a model along the path, by one of these models along the path. And then this probability of that model given a free energy is simply given by statistical mechanics, by Boltzmann's distribution. So these are the two important factors. First, we need what is the likelihood of that image being generated by conformation along the path, and the probability of that conformation given a free energy. And then we can generalize this for multiple images. We assume that the images are independent. So we take the product of this probability of a single image given the free energy, that was the expression that I just showed you, times a prior probability. And so using Bayes' rule, we can obtain the posterior probability of a free energy profile along the path given the Cryo-EM images. And so now, once we have this probability, what we do is we're going to generate many samples of different forms of G, different functions of G, and we're going to sample these from this posterior. And then once we sample this, for example, with a Markov chain Monte Carlo, we can calculate its mean and its uncertainty given the Cryo-EM particles. So I'm going to now explain a little bit really how you do this in practice. So there are four main steps. The first step is that you define your path with your M nodes that take you from one relevant state to another. Then you calculate this Bio-EM likelihood that tells us what's the probability of a model along the path to generate an image, and you can pre-calculate this for all the models along your path and all your images. Then what we use is this equation 1 to sample the free energy profile following this posterior probability distribution. So basically, what are the G's? What are the most probable G's given this posterior? And we can do this using a Markov chain Monte Carlo. And finally, once this Markov chain sampling is converged, we can calculate the mean and the uncertainty at each node of the free energy profile. So now I'm going to go quickly into each of these steps. So the first step is extremely difficult. It's basically knowing your system, having some information of relevant conformational states, so structural information of relevant conformational states, and basically taking advantage of tools like MDFF or steered molecular dynamics that take you, for example, from an open state to a closed state in an almost semi-continuous manner. So this is obviously challenging, and for the moment, we will assume that the path that we generate are sufficiently good. However, as a future work, what we want to do is really optimize the path that we're generating. However, I do have to say that this has been done many hundreds of times in molecular dynamic simulation. So we want to build these collective variables, and we have to build a path somehow from one state to another. So then we calculate the Bio-EM probability. So this Bio-EM probability is the integral of this likelihood function, the likelihood of an image given a model from the past. So we already defined the path, so we have a model from the past, and some parameters that take into account all the effects in the Cryo-EM imaging conditions. So these theta are, for example, the orientations of the molecule, the CTF parameters, the shifts in the center, the noise. And this likelihood is simply the exponential of the cross correlation of the image and a calculated image from that model with a given set of parameters. And we assume, for the moment, we assume Gaussian noise. So this basically tells us, okay, this model is very likely to have generated a particular image. So a little bit of the details of the Bio-EM algorithm, and it's basically that we perform 2 rounds of, say, refinement. The first round is an all model rotations, all model orientations against all observed images. We do this in CUDA. And then we have a second round that basically takes the best rotation, so say that here this box is the best rotation for this image, and we zoom over this box and we refine the orientation search. So basically we're looking for the best pose that help us calculate this posterior probability. Okay, so now we calculated all the probabilities, the similarities between each model and each image. And then what we do is we do a Markov chain Monte Carlo. So here, say that I choose one of these. We have a starting free energy profile. So this is our free energy profile. This is our path. And then I choose randomly one model along the path, say that I choose this one. And so what I do is I attempt a displacement of the free energy. So I'm just going to, for example, shift up the free energy. And then what we do is we use Metropolis algorithm to accept or reject this free energy, given the posterior probability that we calculated before, defined by the equation 1. And here we're also including a prior to smooth the free energy profile for it not to be very rigid. So we do this many times. We attempt many samples of this posterior. So say here in green, here are the posterior samples. So we attempt many displacements. We only accept the ones that tells us with our probability distribution. And basically we run this until convergence, and we run it for several independent replicas. And then this gives us a possibility to calculate a mean, an expected value for each node, and also error. It also gives us the uncertainties. What is the uncertainty around this point for the mean free energy at that node in time? And we can, if we do like our benchmarking, we can compare this with an underlying free energy surface that is given. And this is something that I'm going to show you with the synthetic data. Okay, so what are the advantages? The first advantage is that we can build a complex one-dimensional path in a multi-dimensional space that can take into account large conformational changes, non-trivial changes in the biomolecule. The second advantage is that an image doesn't have to correspond to a single node along the path, a single model. So there's a probability that Bio-EM, what it does is it tells us, okay, there's a certain probability along the path that the image corresponds to these nodes, but it's not like a direct assignment. The third advantage is that with our Bayesian formalism, this probability naturally weighs the contribution of each image. So basically, and this is something that I'm going to show you further on, images that have low signal to noise ratio, that are, say, bad, they will receive a very low weight and will not contribute so much to the averaging and analysis. Okay, so now I'm going to talk to you about some results for synthetic data. And the questions that we want to answer are, can we recover the free energy profile using this methodology? And if so, under what conditions? What are the Cryo-EM imaging conditions that limit its recovery? So here we have the simplest system. This is a one-dimensional movement of an HSP90 chaperone that is involved in many folding process. This is a V-like shape, and you have like a closed conformation and an open conformation. And we are following ideas to generate the images from recent work by Frank's group. So how do you generate reasonable and good synthetic Cryo-EM images? So here, what we have is, this is the synthetic free energy profile that we impose. And here we have our path collective variable that goes from the closed state to the open state. So basically here, to give you a little bit of intuition, if you're in a well, what this tells us is that these conformations here will be much more probable, which means that they will appear many more times in the images. And if you're in around a barrier, what this tells us is that this conformation is very unlikely. It will not appear many times in your data. So this is our underlying synthetic free energy profile that we impose on the Cryo-EM image distribution. And we use two orientation round estimates of Bio-EM. We generated around 13,000 particles, and we have uniformly distributed orientations, defocus, and a very wide range of signal to noise ratio. So we're going from 0.01 to 0.1, where what is estimated nowadays for Cryo-EM data, we're around this, for real Cryo-EM data, we're around 0.1. So this is an example of the images that we generate. So here what I'm presenting are the Cryo-BIFE results for two orientation rounds of Bio-EM, so two orientation round estimates. And note that the first round is not so good. It's like a coarse estimate of the pose. So in black, you see the underlying free energy. In orange, you can see the results from the Cryo-BIFE method using all of these particles for the first orientation estimate. So you can see that it's not so good. We kind of identify the wells, but we're way off the activation barrier. However, if we have a better pose estimate, then we can recover relatively well the free energy profile. So we can, the wells and the shape of the wells is much better, and we're much more closer to the knowing the true activation barrier. So something that's very interesting here is that here we have a very wide range of signal to noise ratio. So why does this work for such a wide range? And so the reason is the following. So here, what I'm graphing is the signal to noise ratio of each image versus the log of the posterior probability of that image given the true underlying free energy. So here, what you can see is that the images with low signal to noise ratio, those that have a lot of noise, do not weigh so much and do not contribute so much for the posterior. So their weight is much less. And so having images in a set that have high signal will improve the free energy recovery, because these, the ones that are here, will weigh much more and contribute much more to the posterior. So this is also shown in these results. So here what I did is I grouped the particles and I separated in two classes. So here in green, we have the low signal to noise ratio. And basically you can see that if we only have particles between 0.001 and 0.01, basically the recovery is really, really bad. You have very large uncertainties and it's really difficult, like it doesn't work. And for the high signal to noise ratio, which is the cyan curve, you can see that the recovery is almost perfect. You have spot on the barrier, the shape, everything is very good. But what's interesting, and this is like the point that I was mentioning before, is that the blue curve, which is using all particles, combining both the low signal to noise ratio and the high signal to noise ratio, you can have a good recovery, a relatively decent recovery, if you put all the particles together, because the ones with high signal to noise ratio will weigh more, will contribute more. So adding bad particles does not really hinder the free energy recovery if there are some good ones in the set. Okay, so the final like assessment that we did for this very simple system was the number of particles. So here we randomly picked 3.3 thousand, 6,000 particles and all particles, and you can see that basically with very few particles we can recover the underlying statistics of these free energies. But if there are too few particles, what happens is that you have large uncertainties here at the barrier point. So you have large error bars where there are very few populated states. Okay, so what happens for more challenging systems? Actually, we tested the method over many synthetic systems. But here I'm just going to show you one example. So this is molecular dynamic simulations of an hexapeptide in explicit solvent. And this hexapeptide is charged in its oppositely charged in its extreme. So it has two conformations that are preferable, either the closed conformation or the open conformation. And the nice thing is that we don't need extremely large supercomputers to really have a full convergence of the conformational space of the peptide. So we took random snapshots from the MD. We generated synthetic images from these snapshots following the same ideas. And so we ask ourselves, does the free energy recovery from 2D projections with low signal to noise, is it equal or similar to the recovery if we use the trajectory, the 3D conformations from the molecular dynamics simulations? And so here, this is what I'm graphing. This is the free energy. And in green and in black, this is the free energy extracted from the 3D conformation from the molecular dynamic simulations. And in pink, you can see the results from our Cryo-BIFE method. And basically, we can identify the metastable states of the system, the two minima that are here, and we can also nail the activation barrier. So basically what we've learned, unfortunately, I don't have time to show you more systems, but what we learned for these synthetic systems is that for realistic cases, we can recover the free energy profile, even if the conformational changes are low or small between conformations. Orientation accuracy, as I showed you, and signal to noise ratio play key roles in really recovering a good free energy profile. When we can control the orientation accuracy, we can always refine our search, but the signal to noise ratio, well, with the experiments, we can definitely try to get more signal. And the last thing that I want to mention is that this probability naturally weighs the contribution of each particle without need of hard filters of classifying out particles. So particles with poor information due to low signal to noise ratio, bad projection direction, large defocus will naturally have a low posterior. Okay, so now I just want to finalize the talk showing you some results with real Cryo-EM data for a membrane channel. So this is a calcium activated ion channel, and it was solved at high resolution by Yingfang Cheng's lab a couple of years ago. And so basically they have these two conformations, the calcium bound conformation and the calcium unbound conformation. So we focused on the set that was available at Empire with calcium, that was made with calcium and imaged with calcium. And this was the set that was used to generate the high resolution structure of the calcium bound state. And something interesting is that, and I think this is very standard in Cryo-EM, is that you start with a large number of particles. So here they started with 1.2 million, and they do their classification, and they make the high resolution structure, obtain the high resolution structure with less than 15% of the data. So we have several questions that we want to address. The first question is there a small population of the calcium unbound state, so the other state that wasn't used to reproduce the data, in this set? And why do we think this? Because of biochemistry, equilibrium populations, not all in equilibrium, not all the proteins will be calcium bind. At least this is what we expect. And the second question is, can we extract a free energy profile from going from the calcium bound state to the calcium unbound state? What does this free energy profile look like? Is it reasonable? So how do we create the path? So what we do is we use steer molecular dynamic simulation. So this is our ion channel. We include an explicit membrane and solvent and ions. And then what we do with these steer molecular dynamic simulations is that we bias the system. We put a fictitious force over the system to go from one state to a reference state. And so then to select the nodes along the path, what we use is we use the RMSD with respect to this calcium bound state. And we also include a membrane belt that mimics the detergent in the experiment. So here, what I'm showing you just a little bit more detail. So this is the C alpha RMSD. These are the paths along the node path. So the conformations that we chose for the path. And in purple, you can see the RMSD to the C calcium bound state. And in green, the RMSD to the calcium unbound state. So what you can see is we're going closer to the calcium bound state, and then we're going further away from it, and then we're reaching much closer to the calcium unbound state. So this is just a very short movie of how the path nodes look. And so basically something to note is that really the conformational changes here are not dramatic. We're talking about 2-3 Armstrong differences. Okay, so here I'm presenting the Cryo-BIFE results for real particles. So what we did is we took the particles from the entire set and we randomly picked 15,000 particles. So just 15,000 out of the 1.2 million particles used for at the beginning. So not those for reconstructing the high resolution structure, but the entire set. And then we ran the Bioem and Cryo-BIFE analysis exactly before, as we did with the synthetic data. So here we have our path collective variable, and this is the free energy. And we can find 3 metastable states. The lowest free energy minima corresponds, as we expected or as we hoped, to the calcium bound state. This is the state that was used, basically the particles that were used for the high resolution reconstruction. But we can also find the second metastable state is the calcium unbound state. So this state represents also a local free energy minima. So basically this is telling us, yes, in your data, you do have the other state. And we also found this other small metastable state. We actually don't know what this corresponds to, but also something to take it like in mind is that for turnover processes in equilibrium conditions, we expect barriers. If we see conformational changes, we expect barriers of the order of 1 or 2 kBt. We don't expect something of 10 kBt. Okay, so this is basically what I wanted to show you. These are the conclusions. We have developed a path collective variable and Bayesian method for extracting free energy profiles using Cryo-EM particle images. And this formalism allows us to weight the contribution of each particle and extract a mean of the free energy profile and its uncertainties. And the free energy recovery is possible under many realistic Cryo-EM conditions. So this is some future work. So something that we want to work on is how to optimize the path. So if you have many paths, for example, that take you from one state to another, how do you choose the best one? So, and how to optimize this, say, automatically in the future. And also, if we are looking for applications over more challenging systems. So if you have a system that you think we could apply this on, we would be really happy to collaborate. And this is also a motivation for the community. So the motivation is let's try to use more data. Let's not throw so much data away. And let's try to image and do the cryo-EM imaging conditions at more ambient conditions. So the temperature pre-freezing, let it be more ambient temperature. The calcium concentrations or the ion concentrations, let it be at more, even more. We can go even further and go further to more realistic biological conditions. So thank you very much for your attention. I want to thank also my students that have done their work here. There is Julian, which is looking for PhD positions. He's finishing his master's soon. And I also want to thank our collaborators from the Flatiron Institute. And I also want to do some advertising. We will start a structural and molecular biophysics group at the Flatiron. And so there are postdoc positions available. So please contact me. And I'm happy to receive questions. Thank you very much.
All right, thank you very much, Pilar.